Hello boys, welcome back to another session of video tutorials, and this is meant for class 12th biology students, and this is for 17th October 2020. And in this session I am very specific with a concept called DNA fingerprinting. Though yesterday we talked about it, various steps involved in that, but still I wanted to have a repetition of this because this is very important concept, not only for your CBSE board exam as well as your NEET exam. It's also very important for your future. Hence I am just going to repeat this once again. So what is DNA fingerprinting? DNA fingerprinting is a way to identify certain individual rather than simply identifying a species or a particular trait. A technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. We are going to use DNA. And what are the various steps involved in that? Let's see what are the various steps involved. 1. The process begins with a blood or cell sample from which the DNA is extracted. So we are going to collect either a cell, maybe it is a skin cell, maybe it's a bony cell, even a cell of a nail or blood. So we extract the DNA. Step 2, the DNA is cut into fragments, as you are seeing, using restriction endonuclease enzyme. The fragments are then separated into bands by a process called electrophoresis through agarose gel. That's second step. Third, the DNA band pattern is transferred to a nylon membrane. And fourth, a radioactive DNA probe is introduced. The DNA probe binds to specific DNA sequences on the nylon membrane. That is the purpose why DNA probe is used. Step number 5, the excess probe material is washed away, leaving the unique DNA band pattern. The radioactive DNA pattern is transferred to X-ray film by direct exposure. When developed, the resultant visible pattern is the DNA fingerprint. So finally we will get this DNA sequences, nitrogenous basis sequences in this form. So this is what basically we are doing it. And who invented it? As I said yesterday, thanks to this gentleman Alex Jeffreys in 1985, the greatest discovery, and we must salute him. And what are the samples that we require for DNA fingerprinting? As I said just now, it may be blood, it may be just one hair follicle, one drop of saliva, small amounts of semen, body tissue cells. The cells may be skin cells, the cells will nail, there can be hair. So DNA samples have been obtained from even vaginal cells transferred to the outside of a condom during sexual intercourse. So any of this is enough for us to do DNA fingerprinting. What are the steps involved in that? There are 8 steps for DNA fingerprinting. Step 1, isolation of DNA. So keep a notes and write down all this. These are very important. Isolation of DNA. DNA must be recovered from cells or tissue. Only a small amount of blood, hair, skin is needed to isolate DNA. So this is how we do. This is the process. Okay. And then step 2, digestion of DNA by restriction endonucleases. DNA is cut into fragments, as you are seeing, using restriction enzymes. Each restriction enzyme cuts DNA at a specific base sequence, like you are seeing here. See, at a specific base points, it has got the ability to cut. And then the sections of DNA that are cut are called restriction fragments. This yields thousands of restriction fragments of all different sizes because the base sequences being cut may be far away, long fragments, or close together, short. So we may get the mixture of long fragments and short fragments. Commonly used restriction enzymes are HAE3, HINF1, ALU1, ALU1. So reaction mixture is incubated overnight this is compulsory, at 37 degrees centigrade. And step number 3, that is electrophoresis. So electrophoresis makes the agar gel, and of course the agar gel is very important. Fragments are separated on the basis of size. Just now we said long size and short size using a process called gel electrophoresis. And the DNA fragments are injected, as you are seeing here. They are injected into the into wells, and an electric current is applied along the gel. You can see the positive and negative, and DNA being negatively oriented, it moves towards the anode, and the shorter ones will travel fast. So short fragments of DNA will be moving fast towards the anode. That's what we are going to see in the next slide. As I said, yes, see, look at this. DNA is negatively charged, so it is attracted to the positive end of the gel. The shorter DNA fragments move faster than the longer fragments. So thus the DNA is separated based on the size. That is the purpose why we are using the gel electrophoresis. And agar gel is very good in that. It will easily separate the DNA fragments. And step number 4, transfer of DNA on nylon or nitrocellulose membrane. Now whatever the bits pieces are there, well, the DNA fragments are transferred to a nylon sheet, as you are seeing, on the gel, and soaking them overnight by the process called southern blot. So here you can see the vacuum, agar gel, okay, nylon membrane, Wattman paper, rubber gasket, and porous mat. This is how the southern blot technique will take place, and southern blot will help us in making the probe labeling. Adding radioactive or colored probes to the nylon sheet, which is complementary to target sequences. Each probe only sticks to 1 or 2 specific places on the sheet, as you are seeing. So almost we are getting the barcode like structures. Now step number 6, hybridization. Labeled probe DNA should be hybridized with the complementary sequences located on nylon membrane for the detection of position later, like this. Membrane is washed to remove non-specific binding and clearing of the background. And once we get that, now we go for the step number 7, autoradiography. To detect the sequences in genome bound with the hybridized radioactive probe on membrane. Technique involves alignment of hybridized membrane with X-ray film. X-ray film, after its development, shows multiple number of bands that looks like barcodes and known as DNA fingerprints. So you can see the fingerprints and how to interpretation of band patterns, analysis of band patterns of different individuals. Like for example, a suspect, somewhere murder has taken place. So we have collected, let's say, the blood samples there on the spot. Now from the blood sample we get the DNA, and we use the above 7 steps. We get that band pattern, and then police department will ask questions in the area and collect the blood from the suspected people, or maybe something is available in the police station records, so they match that. So different people's pattern, analysis of band patterns, comparison of position of bands, so where exactly the bands are there. So this is bands. Computer software are also available for the analysis of DNA fingerprints, so comparison will be there. So what are the use of DNA fingerprints? Diagnosis of inherited disorders. This is the greatest help. Helps diagnose disorders in both parental as well as newborn babies. Disorders may include cystic fibrosis. We can find out whether the person is having cystic fibrosis or hemophilia. We talked all these are genetic disorders. Huntington's disease, and then Alzheimer's, the very dangerous one, sickle cell anemia, and thalassemia, and of course even myasthenia gravis. All these can be identified. And then in the crime scene, Forensic science is the use of scientific knowledge in a legal situation. The DNA profile of each individual is highly specific. The chance of two people having exactly the same DNA profile is very rare, 30,000 million to 1, except for of course identical twins. We can identify the culprits, and then the pattern of DNA profile is then compared with those of the victim and the suspect. If the profile matches the suspect, it provides strong evidence that the suspect was present at the crime scene. It does not prove he committed the crime, but of course he was there, so there can be some clues. If the profile doesn't match the suspect, then that suspect may be eliminated from the inquiry. So it will be of great help to police. And for example, a violent murder occurred. The forensics team retrieved a blood sample from the crime scene. They prepared DNA profiles of the blood sample, the victim, and the suspect. And it is as follows. Look at this. This is what I said, barcode. So this is the DNA fingerprinting profile of a suspect. This one. And blood sample that is available in the crime scene. This is the one. And the victim's profile is this. So whether the suspect's profile is matching with the blood sample from the crime scene. Yes, look at that. Look at that. Look at that. So, will the police come to a conclusion easily? That is the greatest use of this. And medical problems, DNA profiles can be used to determine whether a particular person is the parent of a child or not. That is paternity. A child's paternity as well as maternity can be determined. Who is father, who is mother, biological father. Okay. This information can be used in paternity suits, inheritance cases, immigration cases, all this. So again, on paternity test, like many claim, maybe because of property, because of position, because of power, people claim that so-and-so is father, his father, or mother, his mother. So just by claiming in social media, they cannot. There has to be a DNA fingerprinting test. So if the person and whom he is claiming to be either mother or father, if the DNA matches, then only there can be. So by comparing the DNA profiles of a mother and her child, identify DNA fragments in the child which are absent from the mother and must therefore have been inherited from the biological father. This is how we come to conclusion. Is this man the father of the child? Now, mother, this is the profile, DNA profile. This is the DNA profile of the child, and this is the man whom the child is claiming to be the biological son. So if you see, whatever is missing here, is this present here? Now for example, child and the father, look at this, and look at this, and of course look at this. So there can be plenty of chances. And so uses of DNA fingerprinting, personal identification. The US Armed Services are just beginning a program where they collect DNA fingerprints from all personnel for later use in case they need to identify casualties or missing. So there are n number of places where this DNA fingerprinting can be used. Hence, this is very important concept, and who knows, some of you may become experts in DNA fingerprinting in future. All the best.